YouTube.
He's going to start a live it. He's going to live it on the YouTube.
Okay,
So,
okay.
Okay. Hello, everybody. Welcome to this special program, a webinar by Department of Botany at Central University of Punjab. We are having this webinar in connection with UN International Day for Biological Diversity today, 22nd of May, 2021. So here with us, we have our esteemed Honorable Vice Chancellor, Professor Raghvendra P. Tiwari, sir. Tiwari sir is supporting all of our endeavors from the right in the beginning. This particular event, we have also an international quiz competition after the presentation. Our chief guest of today is Dr. Manoj Prasad. He is a scientist 7. He is from NIPGR in New Delhi. So his talk, and after his talk is going to be the international quiz competition. So please stay tuned. And at the onset of this particular program, I wish you all a very happy International Day for Biological Diversity. So let me invite our president, our vice chancellor, to deliver the presidential address and to welcome our chief guest. Over to you, sir, Professor R.K. Tiwari.
Dr. Manoj Prasad, eminent scientist, Professor Sanjeev Thakur, Dr. Felix, Dr. Vinay, and all those participants and students, scholars, and faculty members of Central University of Punjab. First of all, my greetings to all of you on this occasion of Biodiversity Day, and also greetings to all those who are surviving the onslaught of corona pandemic. And we are, of course, the highest form of biodiversity. So if we survive, then all other forms of life, we can ensure that they also survive. So our survival is of utmost importance. This is general thought, this is traditional thought. But if we see our activities during the last 2-3 decades, and the type of discourse that we have had with the nature, and will with all forms of civilization, all forms of all aspects of life, we find that the biodiversity per se is at great danger. I'm now, in fact, currently I'm writing a narrative of my village, and the title of the narrative is that, "Can someone return my village?" And under that, there is a separate chapter on the floristic diversity of my village. And during the last, 4 decades, now it remains maximum 5%, what it used to be. Every household had around 10 types of trees. I'm not talking about the number. 10 types of trees, every traditional trees like, banyan tree, then neem, babool, bear, and some orchid tree, etc. Every household has, and had a kitchen garden. Then there used to be orchids, like my own grandfather had big mango orchid, and there were around 130 mango trees. Now in that orchid, only 10, not even 10 now, because recently there was, because of the cyclone, some tree fell. So, the maximum 5% is now preserved. And this is the case with almost, every part of the country. There are certain regions, certain attributes attached to those areas which are very rich in biodiversity, and these areas we call hot spots. I have worked in Northeast for 32 years. And then, so Northeast is big, mega biodiversity, that is within hot spot, of course, mega diversity rich zones. Then we have the Western Ghat. And you see, one of the main reasons why these two areas is still, host large amount of floristic diversity and faunal diversity is because these are not easily accessible and human interference has been minimum in these areas. This is one. Of course, if you talk about the biodiversity of Northeast, what we find is that it is because of the land connection that was established between the Indian plate and the Burmese plate about 20 million years, and then there was floral exchange, and then proliferation of flora, and then through evolution, newer types of species have emerged. So that, you see, and that is still there because the per kilometer or the population density, if we say, it's very thin as compared to other parts. Kerala, it is very rich. I'm told that if all the people of Kerala return to Kerala, then they will not have enough space to lie down also, forget about having houses and all. So much population density. So, so that, wherever there is thin density of population, number one, and less interference with the nature, biodiversity is still preserved. But, see, as we know that the number is increasing, and more so with Indian subcontinent, where, the maximum population, say we can say that around 40% of population lives in the Indian subcontinent, including Pakistan, Bangladesh, and some Indonesia and some other areas. If we see the greater Indian subcontinent, I'm not talking about politically, but geologically, when we consider the Indian plate, as a tectonic block, and that has in fact also led to the rich biodiversity in the Himalayan region. This collision of Indian plate with the Eurasian plate and all, and intermingling of fauna and flora. So this is there, but now because of increased human interference, and because the population is increasing, and the land area where available for survival of mankind is also shrinking. Therefore, we are enclosing the forest and all. This is there. But if we see the total contribution of flora and fauna, and how they preserve the environment, and how they are responsible for maintaining conducive environment, and suitable climate for survival of the humanity, then we really think that we are really in very pathetic situation. Now, how to reverse this, and this this has actually become the mass movement. And I would like to pay my tribute to the great environmentalist Sundarlal Bahuguna, who passed away yesterday, and he launched very touching and movement, where that captured the imagination of every villager, Chipko movement. So something like this has to happen, and the great contribution made by Bahuguna ji, this should always remain in our mind, and for preservation of not only preservation of what is done, but also the plantation as much as we can do, and that is what we are doing with great support of our faculty members. We are doing the university campus. Recently we have set up this Vatika, where Griha Nakshatra, etc., their trees correspond, their trees are planted, and we are planning for massive plantation drive of not only commercial plants, but also fruit-bearing plants, which can really attract different types of birds, avian, and then that really becomes alongside Homo sapiens, all other forms of life, particularly the avian flora, coexist in the Central University of Punjab campus. That's our endeavor, and occasions like this, of course, today is very important occasion, and we should talk about it. But the preservation and creation of biodiversity, this should always remain engraved in our mind all the time, 24/7. Then only perhaps we can maintain the biodiversity, great biodiversity that the nature has endowed. I don't know, because this has been one of our core strengths. If we see this Indian civilization discourse, we find that we were very close to nature, and we respected all forms of life, including plant life. Still, in villages, the trees are worshipped like God. Banyan trees worshipped, people neem, amla, and everything, all, important plants which give, medicinal fruits, and not only fruits, but the different parts of the plants can be used for cure of various types of ailments. And, I always say that if we say normally who is the most faithful creature on this earth, we say God. We say dog, that this dog is the most faithful creature on this earth. But I say most faithful creature on this earth are plants. If you cut the branch of plant will still continue to give oxygen to you. But if you don't give promotion to any teacher, he will start, harming you. See the plant. You can cut everything. If you leave even one or two leaves, leaf will always give you oxygen. You have cut the entire part, 95% of the part of the plant you have cut. If you have leave certain leaves, so without bothering for their life, they're giving oxygen to us, which is very important. Now we realize the importance of oxygen in corona pandemic. So in a sense, what I'd like to say is that if you want to see that my grandchild or his grandchild survives on this planet Earth, then we have to, plant more and more trees, and we have to ensure that the oxygen level in the atmosphere is the same that I inherited from my father. The same amount of oxygen level in the atmosphere, I should transfer the button to my son, and so on and so forth. This way only we can ensure this generation after generation. We all know that Felix is always very active, and he knows all the days in advance, and he plans for good activities, good academic activities on occasions like this. So my compliments to him, my compliments to Botany Department, Dr. Sanjeev Thakur, Vinay, and all those who are in this platform, and of course to Dr. Manoj, who has very kindly agreed to deliver an important talk, deliberation on this very important topic, on which the future of not only India but the entire world lies. So I welcome you, and I am so thankful to you for sparing valuable time for having interaction with our faculty members and students. So with these few words, my compliments to again for Botany Department and to Felix for having organized this seminar. Thank you. Jai Hind, Jai Bharat.
Thank you so much, sir, for your kind words, and really, to the point. And yes, so you have highlighted the Sundarlal Bahuguna, and we missed him only yesterday. The incident happened. And yes, thanks a lot for your kind words, sir. And in your able guidance that the university have made several, you have already pointed out our Nakshatra Griha garden, which has been inaugurated only very recently, 5th of May. And yes, it's your initiative that actually made our entire university campus plastic free. I would like to highlight that point. It's completely plastic free and also smoke free. Smoking is disallowed in the campus. So a lot of interesting policies that you're actually bringing to our campus for the biodiversity conservation and promoting the sustainability and zero waste movement as well. And yes, so we are really thankful to you, sir. And it's in your able guidance that Department of Botany also recently got this FIST grant, DST FIST grant. And thanks a lot for your great support. And also our herbarium has been, enlisted in the New York State Botanical Gardens Steery Herbarium, the index herbarium. And now, yes, so without wasting much time, I would like to invite our esteemed guest, and those who are watching this event live, please stay tuned. And after this event, we are going to start the quiz program. Okay, so first is talk by Professor Manoj Prasad, sir, and allow me to share a screen. I will just introduce him. Well, words are not sufficient. I'm not even qualified to introduce him, but I will try to introduce him. So Dr. Manoj Prasad is J.C. Bose Fellow. It's J.C. Bose is one of the prestigious award by the DST, by the government of India, at NIPGR, in New Delhi, one of the prestigious institute dealing with the genome resources, And he is the fellow of all the 3 academies at the young age to become elected as the fellow of the 3 academies. It's a big achievement. Congratulations, sir, for this great privilege. And he has got a large number of awards, including Golden Peacock Award, Tata Innovation Fellow, National Bioscience Award, Hiralal Chakravorty Memorial Award, and many more. Of course, a large number of awards he has got. We have already shared his profile with the participants, so please have a look. And he has published more than 160 high impact peer-reviewed international publications. And he has been working on neglected millets named foxtail millet. So that is part of our ancient, Indian ancient the food. Of course, I hope you will be talking a little bit about the millets. It's kind of completely neglected in today's world of this fast food and Western culture. And Professor R.P. Tiwari sir also highlighted that why we are forgetting. So we should not forget what our forefathers and foremothers have been telling us to be, to live a life which is as close to nature as possible. And his research area is in plant molecular genetics, genomics, and stress biology. Some of my colleagues are working right on this subject, and I hope all of you are here. I can see Professor Sanjeev here. Hello, Dr. Sanjeev. And Pankaj is also here. So you both are working areas very close to what Professor Manoj is working. So we will have an extended discussion after his first presentation by Dr. Manoj. Over to you, sir. You may please start your presentation.
Okay. So my screen is out of view?
Yes, sir. All is well. Go ahead, please.
Okay. So good afternoon, everyone. It is a nice meeting you all through this virtual platform on the eve of International Day of Biodiversity. And my foremost thanks are to the Honorable Vice Chancellor, Professor Raghavendra P. Tiwari, for giving me this opportunity. Sir, I heard your introductory lecture, and then I was really finding is difficult because you have told most of the things that is in nature we are encounter with day-to-day life. I also like to thank Dr. Felix Blast, Associate Professor and Head of the Department, and Dr. Vinay Sharma, Assistant Professor of Department of Botany, for inviting me to deliver this talk amongst the distinguished audience today. As you know, the nation or the entire world is facing such a difficult situation due to the pandemic spread, but we proved that our academia is unstoppable, and we keep organizing such online programs to keep the scientific community engaged in active research and providing research updates. I appreciate the participants also who are gathered to prove that nothing can stop us from learning, and I hope this virtual public talk provides you an updated information on the roadmap for outcome-based education and research. Today, my topic has relevance to the ongoing issue of hunger and malnutrition as the forthcoming challenge of how we are going to feed 10 billion people by the year 2030. So basically, my talk has 2 parts. One is to show you the challenges ahead of us, and that is how we are going to feed the people. And the second is the answer to my question, where I will show how mainstreaming biodiversity and deploying biotechnology will address the food and nutrition security. So to begin with, let me introduce what is the importance of biodiversity. So the biodiversity forms the basis of life. Therefore, it is important that we preserve biodiversity not only for our survival, but also for our food, nutrition, and livelihood security. Also, biodiversity holds sustainable solution for adaptation to climate change, and we should always remember that we are not the only species on this earth. We share this planet with hundreds of thousands of species with equal rights. Therefore, we must learn and educate others to live in harmony with nature, and also to take all the necessary steps to halt any further loss of biodiversity from this planet. As a researcher, we should use this agrobiodiversity by employing all the latest tools of science and technology like genome editing for producing more from less, so that we will save land and further promote the biodiversity on this planet. This will also ensure feeding the ever-growing population, which I am going to explain in my later part of my today's talk. We should also try to mainstream the agrobiodiversity-based agriculture and promote the use of locally adapted crops for food diversity, for ensuring sustainable global food production. As you could see from this picture, wide diversity is present in nature, and that need to be diligently studied. I thank the Central University of Punjab for scheduling my talk in International Day of Biological Diversity. So with this, I am going to the topic of my today's talk. So as you know, the humans are the last one to evolve in this earth, and but we are leading the game in terms of number. As of yesterday this time, our population counts 7.7 billion, and experts predict that it will reach 10 billion by 2050. To this population, we have assigned three basic things, like namely food, shelter, and clothes. We have been told that without these three needs, survival of the human is not easy. But if you ask whether the 7.7 billion gets the three basic needs, the answer is no. 821 million are still hungry. For every 10 seconds, a child goes from hunger-related causes, and 1 in 4 of the world children suffer from malnutrition, which altogether means that getting food along with will not serve the purpose by getting the nutrition food will. If you see the Global Hunger Index 2019, the Africas and South Asias, including India, are still in the danger zone. You can see here with this different color code. Irrespective of several steps has been taken, including the Green Revolution. Among this, Global Hunger Index score from 2000 to 2019, it was shown that at the regional level, South Asia and Africa south of the Sahara have the highest hunger score of the world, and it has 29.3 and 28.3% respectively. So the global report of food crisis, the GFRC 2020, shows that 821 million people go to bed hungry every night all over the world. And at the new global report on food crisis shows there are still further 135 million people facing the crisis level of hunger or even worse. So this shows that we are still far from a world without malnutrition. And this is the joint child malnutrition estimate that for the child, that 144 million children under the age group of 5, they are stunted. They are too short for their age. 47 million children are under the age of 5 are wasted. That is too thin for their height. And 38 million children are under age 5 are overweighted. So if you see this picture of this joint child malnutrition estimate, 55.9 million children under the age of 5 in the South Asian are stunted. So this situation is not good. Even for 25.2 million children under the age of 5 are of age in South Asia are too thin. And if you see the UN Sustainable Development Goal, out of 17 sustainable goals, 3 are related to food, like then number 1, number 2, and number 3. So when we talk about food, I must say that the world most important botanical garden, Kew, they have estimated that in the world we have more than 11 lakh vascular plants in existence. But that throughout our human history of food, almost 7,000 crops has been used for this consumption purpose. But due to this problem of narrowing down of this food base, now we are coming to 3 major crops, cereal like rice, wheat, and maize currently provide the 50% of the world food. And together with these 3 major cereals, and another 5 crops and 5 animal species provide 75% of the world food consumption. So you can see from 11 lakh to 7,000 to just number 15, the food base is narrowing down day by day. So this is called the great imbalance between the production of a population and the consumption of the crop species. So there is a huge gap between the population and the number of crop species available to feed the population. So you can see here from how we can feed that 10 million people with this only 15 crop species. So it's a high time to focus on mainstreaming the underutilized crop for food and nutritional security. And the second topic of my talk is using how we can use the next generation tools, genomics tools for trait improvement of this crop. Because as you see, the most of the work research has been gone to do the research on the major crops like rice, wheat, maize, and barley. But not much attention has been made for the underutilized crop species. So what is underutilized? They are neglected. And underutilized, neglected means less research attention, as I said, and underutilized means marginally cultivated and used. These underutilized crops, they are cultivated mostly in the marginal areas and by the traditional farmers. And if we want to know what are the underutilized crops, like for example, tubers, legumes, leafy vegetables, and minor millets, they are the different classes of underutilized crops. So underutilized crops that are adapted to marginal environment, they are highly nutritious, and they have the potential for development as the novel consumer products, and they are typically embedded within the local cultural tradition. So these are the features, and they are really very good to come into the mainstreaming of our food basket. So for example, if you talk about tubers, there are several examples: taro, arrowroot, Indian soup, ganola, purple yam, air potato, and elephant food. If we talk about legumes, wing beans, jack bean, bambara groundnut, kidney beans, fava bean, lima bean, cowpea, horse gram, rice bean, velvet bean, and ajusi bean. They are the few examples from the legumes, underutilized legumes. And if you talk about the small or minor millets, they're like finger millet, foxtail millet, proso millet, little millet, Burnett millet, teff, podo millet, fonio, job steer, gunya millet, and brownstock millet. So these are the 3 to 4 different categories of underutilized crops I have talked about. And if I talk about the production of millet in India and also in the state-wise, so India is the second most largest producer of millet after China. And if we talk about the state, the Rajasthan and Maharashtra tops the area of millet production, but yield-wise, Tamil Nadu and Uttarakhand tops the yield in terms of kilo per kg per hectare yield. So coming to the geographical spread of this rice, so millets are already there actually. So it was shown that in a recent paper that rice came little later, almost 4,000 years ago, but millets are already in the cultivation in our food basket. So if I talk about the nutritional potential of millets, this table shows the composition of different important nutritional potential like protein, fat, ash, food fiber, carbohydrate, energy, and vitamin like thiamine and riboflavin. All the millets, they are much superior, 3 to 5 times much superior as compared to the staple crop rice and wheat. So you can see here the values are written, and they are 3 to 5 times much more superior than rice and wheat. So in terms of micronutrient, sodium, calcium, potassium, magnesium, even these are also superior in all the millets as compared to the major cereal crop species. So now millets are growing popularity, and as you can see here that per kg of barnyard millet is costing the farmer 110 per kg. So you can see here, you can get a rice in 60, 70 rupees, but not any of the millets, because production wise, they are very less, but they are the farmers are now exporting because they are getting much more money while exporting this millet. So, and you can see here, millets are also good for immunity, and for this pandemic also, government has also said that you have to eat all the nutritious food, and millet is one of them in terms of nutrition, as I showed in the table. So millets are gaining popularity, and there is one good news that in Madurai there is one variety, that MDU 11, Professor Vanirajan from TNU Madurai, he has made a high-yielding variety of that Banyan millet, and that replaces the rice cultivation in Madurai district of Tamil Nadu. So keeping in view this nutritional potential of millet, our central governments declared that all the millets, as I said, that pearl millet, sorghum, finger millet, all the minor millet, including foxtail, barnyard, little millet, they are declared as nutri cereals by the Gazette notification in 10th April 2018. And following this, the United Nations also declared the year 2023 as the International Year of Millets. So millets are getting popularity day by day, and we need to streamline this thing. And then the question is, can millet address the hunger issue of ever-growing population? Yes, and this world map shows that an integrated map showing the hunger hotspots spanning 55 countries with acute food insecure population by the year 2020, and the small millet cultivation in those regions. So this is a latest one we have formulated in Trends in Plant Science, that how millet can address the hunger issue for the growing population. And what is the roadmap? And we have also postulated a roadmap. For that, we have to identify the millet species suitable for cultivation in the hunger hotspot. We have to identify the region from where this millet is good, which millet is good for which condition. And then we have to optimize the cultivation practices and training of the farmer for millet cultivation. And we have to implement the large-scale production of millet species and optimization of downstream processing. This is very important. And then improvement, that, as I said, that major research has gone to these major crops like rice, wheat, maize, but little research has done. So we have to pay attention for the genetic improvement of millet varieties using modern biotechnological and breeding approach, so that we can get more yield and more resistance. And the supply one, devise appropriate strategies for post-harvest processing, storage, and supply chain. So if you want to know more about this, how millet can endure the food security amidst pandemics, you can read our opinion article in Trends in Plant Science. It was recently published. So as I said, the big threat to agriculture is on its way, because if you see that COVID-19 pandemic, the recession, and 10 billion people to feed, so how we can survive with these sorts of population explosion, recession, and the pandemic like this? So that's why we should focus on the next generation genomics tool, so that these biotechnological tools can be used to improve these underutilized crop species. So to this end, breeding is very slow, because from the early days, breeding is going on and still going on. But to develop a high-yielding variety, it requires almost 8 to 10 years. The first that's high-yielding dwarf, that IR8, developed by the International Rice Research Institute, by crossing with the Beta Indonesian tall cultivar with low yield, with the Chinese DGU gene dwarf variety with normal yield. And you can see the semi-dwarf with good yield IR8 variety was developed in International Rice Research Institute, Philippines. Similarly, there are several success stories of breeding is there, but as I said, the breeding is still slow. So 10 to 12 years is too much time to develop in the present population context. So what we did, we need to expedite the crop improvement program, and how we can do this. But to do this, again, the climate is changing very fast. You can see the prediction that by 2050, the climate temperature will be more than 50. So India has 2% of world's land, 4% of fresh water, but 16% of the world population and 10% of cattle. And the most dangerous point is this: the inland area are much in danger in terms of hotspot for drought and heat and things, something like this. And and in comparison to the coastal area and the hilly areas. Because due to this coastal and hilly area, the rainfall is there, so there is no problem. But in the inland area, the situation is grave, where the most of the food crops are growing. So crop failure is due to the climate change, is, as we all know, that these are the reason: no rain, late monsoon, heavy monsoon, pest and insect and disease, and other natural calamities. So these are the common features for crop failure. And recently you have seen that the pest attack was increasing, that the locust was one of the devastating thing, that these are the natural calamities. No one can stop this thing because this kind of pest can attack anytime. So this short video will show you how it can destroy field after field. And so we have to take care how to control this thing. So according to this, that FAO, most of the damage in agriculture due to this climate disaster is drought. So if you see 83% of this damage of loss in agriculture is due to drought, and followed by volcano, storm, flood, tsunami, and earthquake. So if we talk about the farmers' death, that the National Crime Report Bureau data show that more than 42,000 farmers and daily wages commit suicide in 2019. But this data was before the pandemic. You see, so what is the situation at present? So the take-home message is this: that when all the farmers are gone, then it will be our turn. So we have to be very cautious, and we have to face this challenge of climate. So this will continue, and by 2015, crop yield will decrease by 20%, 25% if we don't address the climate change now. So can biotechnology rescue the agriculture? So this question now, how biotechnology can rescue the agriculture, I will show you some great example. So there, as I said, there are several stresses, and then we can also do biotechnological intervention. So stresses like drought, flood, cold, heat, salinity, radiation, oxidation, and oxidative and stress, and pathogen. This is the biotic stress. And in terms of biotechnology intervention, there are different omics technology like genomics, transcriptomics, proteomics, metabolomics, epigenomics, ionomics, and phenomics. This is the integrated omics technology is required to address this challenge. So if we talk about how biotechnology can help to this challenge, so I can show you that these two cartoons, that two plants, the one is the high yielding, one is the low yielding. So so it means that in high yielding plant, there is the sequence of is really something new or different than the low yielding plant. So in the layman's language, if I say, so if this is the high yielding gene, which is present in this locus in the high yielding plant, then we can transfer this gene from this high yielding plant to the other low yielding plant, and it can be done through genetic engineering. So this is in layman language how one can use the genetic engineering technology to transfer a gene from one plant to the another plant. So in the meantime, the DNA sequencing has revolutionized the biotechnological field. And if you see that there are a lot of genome sequences be done, and these are the kind of noise once the genome sequence data comes, ATGC. So we have to fetch out the noise, and we have to find out the meaning of this noise, and that meaning is this: it encodes genes. We have to identify the genes which encodes for protein. We have to identify the regulatory motifs which identify the control the gene expression. So if you see here, the cartoon shows that we have identified a gene or locus for the high yielding, and this is the additional gene that is known for this high yielding. And we have to clone this gene, we have to insert it into the vector, that we have to put it in the explant, and then through agrobacterial transformation, we can raise the subsequent generation. And by this way, we have can obtain the high yielding variety. So this is the biotechnological tool by using how one can develop a high yielding variety. And to do this, we need a different approach. Like this is the example of agrobacterium mediated gene transfer. And for this agrobacterium mediated gene transfer, we can take a gene from any source. It can be bacteria, it can be virus, it can be humans. So you can take any kind of genes and from XYZ thing, and then you clone it, and then you put it in the plasmid and to agrobacterium tumefaciens, and then you can infect these agrobacterium tumefaciens to the plant, and then you can develop a transgenic plant for your desired gene of interest. Like I have showed you the high-yielding varieties, how can that. So there's a classical example is the BT revolution. And as you see that in the field, worms and insects feed the vegetable parts. You can see there a classical example here shown in the picture. And this BT is the Bacillus thuringiensis, is a species of bacteria, gram-positive bacteria that lives in the soil. And it makes protein that are toxic to some of the insect when it eaten and the insect die, but not the others like human. If the human take, they will not die because that is not the thing, because for the BT that required the alkaline solution condition for its activity, but in human we have an acidic in our gut. So this is the structure of this BT gene, actually Bacillus thuringiensis, and this is the BT toxin crystal. So when the insect bite this leaf of containing this BT crystal, it dissolves in the high pH insect gut and become active. So in the insect gut, the alkaline the condition was alkaline, so the BT toxin easily dissolved, and it can make a pore in the gut of the insect, and the spore become so that the insect stop eating and due to starvation it die. So by this way, from a bacterial gene, we transferred it into the plant, and this has made the revolution for the BT thing. So the gene is the cry gene, the crystalline gene. It has been cloned, and then it was through agrobacterium tumefaciens we can transfer it to the plant. So this is the example of this BT crops, and that is, you can see here the non-BT. There's a severe attack, and this is the BT brinjal. Similarly, non-BT cotton and BT cotton. And see the non-BT cotton corn and the BT corn. So this is one of the classical example how biotechnology helps to do the yield loss. So I have shown the bacteria virus to bacteria to plant. Now I can show you our own example that how plant to plant gene transfer can be done. So to do this, what we have done, as I said, that we work on foxtail millet. The whole genome sequence was done, and through annotation, we have identified 586 genes, which were functionally annotated to responsive to water. And further annotation has been done, and we have identified certain candidate genes like small Hitchcock protein, aquaporine, MIP transcription factor, Werke transcription factor, and ADP ribosylation factor. So these are the candidate genes we identified through this annotation process. And I'm going to talk about now in very layman language how one gene we have transferred from C4 foxtail to C3 rice plant. So as I said, the rice grows at 28 degrees centigrade, 24 to 28 degrees. But this foxtail millet plant, the C4 plant grows until 50 degrees centigrade they can tolerate. So there is a huge difference between growing temperature between C4 and C3 plants. So foxtail is a C4 and rice is a C3 plant. So we identified one Hitchcock protein 27 in our Citeria italica foxtail millet, which is only present in the C4 pandicoid species, not in any of the C3 grasses like rice and wheat. It's not present. So what we did, we overexpressed this HSP27, and you can see from this transgenic lines that this is the control one. It's almost it was dying, and these transgenic lines they can happily grow till 50 degree temperature and shows enhanced tolerance to heat stress as well as improved agronomic traits such as plant height, tiller number, panicle number, and size, number of seed per panicle. So there is no yield penalty, and it can grow till 50 degree temperature. So what is the mechanism behind this? So in simple, simplified way, I can say this is the normal gene, and this is the normal protein. In extreme heat, this normal protein can denature. So that's why there is no function and the plant die. In case of HSP27, this is due to heat, and this HSP27 binds to the other protein and prevent their degradation. So that's why the plant happily can leave. So this is a simple way, but this binding activity of HSP27 with other proteins, to do this, we have done a lot of biochemical, physiological, and molecular biological experiment. But due to the time constraint and simplicity, I have not talked anything about this to make it very simple. And that's why I have told that HSP27 binds to other protein and it prevents the degradation of this due to heat. That's why it can function normally. So for crop improvement program, now we have 5 Gs. 5 Gs means genome. Genome means we have to sequence the genome to develop genomic tools and resources. We need a germplasm collection. For any crop improvement program, we require a very good, rich, diverse germplasm. We have to identify the genes and to delineate the function of the gene. And to do this, we need genetic engineering to develop this this high yielding and desired phenotype, and gene editing like CRISPR-Cas9 technology, which can also precisely edit the target gene of our interest. So these are the 5 Gs for crop improvement program is required for any crop species. So now I'm going to show you how we develop integrated pipeline for gene family analysis, because why gene families are important to characterize, because we have to identify the candidate genes for overexpression studies. Because as I said, that for each and every plant species, simple C2H2 type gene finger transcription factor in foxtail, there are 124. We cannot characterize 124. We have to pinpoint which is the most important one. Like I have showed you the HSP27. In foxtail millet, there are 113 HSP genes are present, but we cannot characterize 113. But we have to identify a 1 or 2 candidate gene which is differentially expressed and different than other HSPs, so that that gene can be introduced to the other cross species to get the benefit. So that's the reason why we do this pipeline for gene family analysis, to curtail down the number of genes or to identify the candidate genes. And by this way, we have identified 110 working transcription factor, 160 cell wall related genes. So there are 3 types of genomic toolkit is available. As I said, that to 5 Gs. So one of these is that overexpression, and in overexpression we overproduce the target protein like HSP27. I have showed you one example that we produce over this HSP27 protein, so that it can, if it has a phenotype, it can show the phenotype by overexpression. We can also do RNAi. We can silence that target gene or protein so that we can get the desired phenotype. We can also do CRISPR-Cas9, that's the mutate the target site of the gene of our interest. So these are 3 different genomic approaches now available to fine-tune the candidate genes to get our desired phenotype. Some of the, other example of overexpression, I have showed you one, that our HSP. Now the second one I'm talking about the autophagy gene, that is cytarated ATG8A, which was overexpressed in rice. This is the gene from foxtail overexpressed in rice, conferred enhanced tolerance to nitrogen starvation. So this experiment was done like this: the 3-week-old seedling of control rice plant and transgenic. This is the control rice plant, 3 weeks old, and this is the transgenic rice plant, 3 weeks old, and grown in a normal hogland solution, and were subcultured in nitrogen deficient liquid medium for 21 days. And then this experiment was done, and it was shown that ATG8 overexpressed in rice conferred enhanced tolerance to nitrogen starvation. Similarly, there is another one that SIASR4, abscisic acid stress and ripening induced protein from foxtail millet. It was overexpressed and also silenced in rice. And this is the drought, and this is the salinity stress, and this is the control overexpressed plant in rice. This is the control plant, and this is the RNAi lines of rice. Similarly, in salinity overexpress control and RNAi lines. And then drought treatment was given for 10 days. And in the third panel, you can see here, drought treatment was given up till 13 days. And then the plant has been re-watered after 5 days. And then you can see the overexpress this gene ASR4. The overexpress this foxtail millet ASR4 are happily growing in rice in terms of drought stress and the salinity stress, but the RNAi lines, they die. So it means that the gene has the function to show that it can tolerate both drought and salinity stress tolerance. So foxtail millet gene has been characterized in rice. So what is knockdown and knockout? I have talked about that overexpression. Now I am talking about knockdown. Knockdown, there are two approaches, RNAi and virus-induced gene silencing, and knockout is the CRISPR-Cas9. So in case of RNAi, there was one single gene, BAHD, that is beat, AHCT, HCBT, and DAT. So these are the four, and this is the first word for BAHD gene of Citeria viridis, causes large stable decrease in cell wall fibrillation and increased biomass digestibility. So through RNAi, it was shown that if we BDHD gene was enhance the digestibility of plant for animal feed purpose. So basically, if this gene was silenced or suppressed, BHD gene, then it enhanced the digestibility of plant for animal feed purpose. Another example of knockdown is virus-induced gene silencing. So virus-induced gene silencing is this, that we want to characterize transiently any plant gene. So the technique shows that the fooling a plant to degrade its own RNA. So to do this, what we did, that that plant DNA has been cloned in the VIX vector, and this RNA virus with this host gene has been transiently overexpressed in the plant, and this recombinant viral RNA through RDRP, it makes a double stranded, and then through DCL, dicyl-like protein, it cuts into form the siRNA 21 to 22 nucleotide, including both DNA and RNA. And this is the normal one, the viral that risk as you cut the viral RNA and at this aberrant RNA. But when it is coming to the plant, because it was integrated or it was embedded in the viral, so the plant doesn't understand whether it is own or it is the viral, so it cuts, and by this way we can silence this this plant gene transiently by using the virus-induced gene silencing technology. So by this way, transiently we can very quickly characterize the function of any gene of our interest. So, for example, VIX in millets are very early stage. As of now, only one vector had been identified for foxtail millet. This is foxtail mosaic virus induced flowering assay in monocot crop. So this FMV foxtail. So for tomato we use the TRV, tobacco rattle virus. Similarly for this cereals, we can use this foxtail mosaic virus, the VIX vector. Now coming to the knockout. Knockout, as I said, CRISPR is that technology is for the knockout, and CRISPR is based on these two two substances. One is the Cas9 protein, that is the scissor that cut the DNA, and another one is the guide RNA. So so the guide RNA sequence matches with the target gene of genomic DNA. So this is the genomic DNA and this is the target gene. And this guide RNA matches the sequence of the target gene. And then this Cas9 assembly binds to the target gene, and nucleus of this Cas9, cut the target DNA, and then DNA repairs happen, usually error prone by two different techniques. One is the homology directed repair, another is the non-homologous joining repair. So by this way, one can precisely edit any gene of our interest. So this is a classical example how CRISPR-Cas9 technology has been used to prevent pre-harvest sprouting tolerance, a major issue in wheat. So in the field, if you see in the field, during maturity, if rainfall there in the field, some of the plants they sprout. They started sprouting in the field itself before harvesting. So that creates the yield loss for the farmers. But by this CRISPR-Cas9 technology, scientist has engineered a single gene, and that is that QSD1. This is the alanine aminotransferase family gene, and this mutation in this alanine aminotransferase gene in wheat, they have make it a pre-harvest sprouting tolerant. So what is this? That as you all of you know, that hexaploid wheat has 3 genomes, A, B, and D. So this triple knockout mutant of this alanine aminotransferase gene has been done, and you can see here both this pre-harvest and this tolerant. There is only ATT has been added in this. So this is addition, not deletion. So addition of this 3 nucleotide makes the plant tolerant to pre-harvest sprouting tolerant. So this is a excellent classical example how this genome editing technology can help the farmers and to make the plant to protect the plant from any disease or thing. There is another classical example of this Solanum pimpinifolium. You can say this is a wild tomato, Solanum pimpinifolium. You can see here the fruit size is like pea, and stress resistance, and the growth is indeterminate growth. But through this CRISPR-Cas9 technology, by editing 6 different genes, this wild Solanum pimpinifolium can be domesticated. So you can see here that same pimpinifolium in the wild, and editing of these 6 important genes like the general plant growth habit, fruit shape overhead gene, fruit size, facilitated, fruit weight 2.2, fruit number, multiflora, nutritional quality, lycopene, beta site. So these 6 genes has been edited, and this domesticated pimpinifolium has been developed by this genome editing technology. So you can see here that in domesticated tomato, determinate growth. This is indeterminate growth. This is determinate growth. Large size, more fruit, 500% high lycopene and stress resistant. So modifying 6 genes of a wild tomato converts the wild into a cultivated species. So this is the beauty of genome editing. All these biotechnological new techniques that have showed a promising future for the mankind. So as I said, that multiomics approach and their application in designing climate resilient crop. So there are different omics technology, like as I said in the beginning, that genome sequencing, genome-wide characterization, gene family, transcriptome, bioinformatics, and metabolomics and proteomics. And through this integrative omics, multiomics approach, we can identify the candidate genes or the determinants, and this can be transferred either through this genetic engineering or overexpression or RNAi or CRISPR-Cas9, and to develop a high temperature drought, soil salinity, and high yield cultivar so that it can be climate smart crop. So in conclusion, I can say that molecular breeding and genetic manipulation have emerged as the 2 most potential technologies for crop improvement. Advances in the next generation sequencing technology have enabled the incorporation of genomics with various disciplines of crop breeding. Functional and comparative genomics have provided the platform for gene discovery and their functional characterization. The key gene or genes regulating a molecular pathway are being genetically engineered or edited to develop phenotypically improved crop lines, and collaborative and concerted research is required to circumvent the global challenge of feeding the 10 billion by 2050. So in nutshell, what I have said today, that biotechnology has a greater potential to address this problem, or today's problem, and advancement of different omics technologies and techniques also help to answer the question precisely. So for the student, this slide is very important, because as you know, a guide or the mentor can only achieve its goal if his students achieve their goal. So I have listed the 7 of my students who have already passed and have been placed very well in different places. So Charu has got everything, all the award, and she is a scientist in NVRI Lucknow. Muthu got the assistant professor position in University of Hyderabad. He also got everything. So all the Foxton millet work has been done by Muthu. Charu characterized the drape transcription factor in Foxton millet. Namisha got the INSA medal for young scientist and Sir Women Excellence Award. She also characterized my other part of the tomato story that I have not talked today, that how host pathogen interaction can help us to develop the resistant tomato variety. Swati characterized the NAC transcription factor in Foxton millet. Pranab also characterized the UPS component gene, SLRPD4, to provide tolerance in Foxton millet. Ashish working on the autophagy gene, and he recently got this Newton Howard fellowship from the British Council. And Avdesh characterized the WD40 protein, he got the Brave Scholarship. So this is all about the student achievement, and more students are working. So I hope that more will get many more awards. So this is the team I have at present, and work in my lab has been directly funded by Department of Biotechnology and Department of Science and Technology. So thank you very much for your patience hearing. I'll be happy to answer any of your questions.
Yes, thank you so much, sir. Could you please exit the presentations?
Yeah.
Yeah, I'm really happy to see your presentation, sir. It is really informative, and it is devoid of sophistication, so that everybody can make out of what you have been doing.
Exactly. Yeah.
Could you please go up? There is an option there, stop presentation. There is a red icon.
Yeah.
Yes, sir. Thank you. And I'm also happy to see Charu Lata there. She's my good friend, and we were together in DST Inspire Faculty Award way back in 2011. And also we both inducted together in INEAS, DST INEAS, and yes, we had one invited talk last year in our department, Professor Kadambot Siddiq.
I know him very well. Yeah.
Yes, he is also doing quite similar kind of work. And I'm really happy to hear a lot of good work you're doing at NIPGR about millet and other, of course, neglected, vegetables. To say a little bit about me, I love this cowpea, ragi, and also the jackfruit, quite neglected fruit, So really happy, sir. And we have tremendous response. Lots of people are watching right now in the YouTube. Lots of questions are there. And before coming to the audience question, so I would like to open the session for my colleagues in the department, as well as others who have joined this. Yes. And thanks, Vinay. Vinay is the one who is behind this, arranging this particular talk. I would like to appreciate you, Dr. Vinay. Over to people, panelists here. So you may please ask the question. So over to Sanjeev, Professor Sanjeev, over to you.
Yes, sir. Thank you, sir. Manoj, sir, it was a very nice talk and really very amazing work, whatever you are doing in this particular crops. So I have a little concern regarding the Punjab. So like you know very well, Punjab has some issues with the rice right now, because groundwater table is going down and down. And so now rice crop is not suitable in the Punjab. So what's the suggestion about this millets? So which millets is the most suitable according to this climate condition of this Punjab region? So may you help me to know much better about this?
Yes. Because I know that I have a collaborator in PU Ludhiana. So they used to grow minor millets and also the pearl millet. So pearl millet is also grown a large area of Rajasthan. But in Punjab also, some places I have seen that pearl millet has been grown because a very high iron content is there in pearl millet. And all minor millets is also growing in a few pockets of Punjab, actually. So I have also field trial in PU Ludhiana for the last 3 years for Foxton millet, and I have got a very good result from there. So because this is a summer crop and it requires less attention, no fertilizer, nothing, and you will get without attention you will get the benefit. And nutritionally is 3 to 5 times more superior than rice and wheat. So we have to change our food habit, because you know that ancient time, I think 30-40 years back, actually, it was widely cultivated before the Green Revolution. And after the Green Revolution, everything changed. People has, or due to taste and whatever you say, that they have concentrated only to this major crop, rice, wheat, and maize. So, but the time has come, because these are the crops, 100 plus crops, they are climate resilient. I can say that nutritionally much more superior. They can tolerate high temperature. They do not require any fertilizer. So what is the problem of growing this kind of 100 plus crops? So that's why in the marginal land, the traditional farmers, they used to grow it for their own consumption. But that is the reason why that barnyard millet or all the millet, you would get all the kind of biscuits in the supermarket, but they are highly priced, because they are nutritionally very, this is good. All kinds of millets are very good for the type 2 diabetic, because that glycemic index in millets is less and below 50, while rice has above 70 actually. So the good thing is this: in all the millets, it presents of good resistance starch. That's why when you use as a carbohydrate to eat any of the millets, the conversion of carbohydrate to glucose is very slow in millet consumption, because due to presence of good resistance starch, the breakdown of this carbohydrate to glucose is very slow. That's why it is good for type 2 diabetic people to consume to the millet crops. And as a contrary, that's why in Tamil Nadu, I know for sure that Tamil Nadu government has given a lot of benefit to the farmers to cultivate this, and they are also cultivating, and then area has been also increased in Tamil Nadu. In most of the southern part of India, you will get ragi dosa anywhere. Even in Aero City also you go, you ask them, because the newer generation, they do not know what is millet actually. So we have to make an awareness campaign so that people should understand, because now we are very fascinating with the fast food and that this kind of fast food, and that they are less nutritious, and they are all are the maida. So it's not good for the health also. So this kind of awareness also is required to provide to the younger generation in this time.
Yes, sir. So in Kerala, we have a new trend. New kinds of restaurants are coming up in cities like Kochi, that that is only about the traditional food, and the food which are ritualistic, and it's very interesting trend. I wish that the millets gets more and more a feature in our the dish in our daily food. We get lots of questions here, and unfortunately there are a lot of confusion about GMOs, and a lot of misconception, I would say. One of the question is, the participant Srikant Swami MB is asking, sir, some people say that GMO plants and the products are harmful for health. Please clarify.
But where is the data? Can you show me one data that the GMO plants are harmful? You never country, Bangladesh, Nepal, these people are using the BT brinjal. We are also using the BT brinjal. Do you have any problem? Yeah, the papaya, you get so big papaya, you would think it's a normal papaya. So these are all underground. We are eating the GMOs actually. Even the maize also. These are all GMOs. But these are all unofficial actually things. But as I said, as a scientist, I cannot say against GMOs, because there is no report, there is no proof that the GMOs has that adverse effect in health. Because that BT brinjal is one of the classical example I can show you. That BT cotton is another example. BT maize is also another example. There are several are in the pipeline, but this is something political decision that scientists cannot take. This is the take by the political party, government. So we are for this, because as I said that 10 billion people you cannot feed with this normal food production. You require something, because this climate is changing very fast. There are a lot of things. this pandemic and then the recession, and as I showed in my slides, there are several hurdles for crop production. Day by day, this area of land cultivation is also reducing due to the urbanization. So how could you feed the people? So that's why I'm advocating always this underutilized crop should be practiced, and it should be grown in the local areas, in the climatic areas, those where barren land also they can use it, because that doesn't require this kind of crop require any kind of attention or any kind of fertilizer. They can also go in the marginal land. That's why that's the very hardy, and they can be as well at the same time they're very nutritious.
Okay, we have one more question.
Sir Felix, can
Yes,
I
Pankaj,
go with the question? Yeah. Sir, very good afternoon. My name is Pankaj. Thank you so much for your presentation. And frankly speaking, it was one of the fantastic I have attended recently. Sir, my question revolves around, because I was very much fascinated by the HSP story, which you have isolated from the pearl millet, and went on to
all millet has that HSP. Because it's a specific.
My question is basically, what made you to decide that HSP27 would be the right candidate for raising the transgenics with the rice? I mean, I would like to understand the reasons which made you, or what was the specific about this 27 which made you to put it into the transgenic as for the rice. I would love to know the behind the story, sir.
Yeah. So as I said, that there are 113 HSP gene in foxtail millet. But after this gene expression study in different abiotic stresses like drought, salinity, heat, and cold, we found that this HSP27 was differentially expressed in all the 4 stresses, but not other genes. So that is the way we, on the basis of expression profiling, we have identified this gene, that this gene can be a miracle because it was differentially expressed in all the 4 different abiotic stress conditions out of 113. So
okay, so my next clarification I would like to have is, sir, this HSP27 is a genotype specific, or was it? It's a random, or it's actually it's a ubiquitous kind of situation.
No. As I said, that for all the millets are C4 panicoid species, and rice, wheat, these are all C3 species. So C4 species are photosynthetically more efficient, and they can survive in heat, more heat actually. But C3 are not like this. So obviously, due to this adaptation that in C4, that HSP27 has been present in all the C4 panicoid species. All the millets have C4, this HSP27, but not a single C3 plants has this HSP27. And we have filed a patent for this also, because we have developed this transgenic rice, and it's going up to tolerate up to 50 degrees actually. You cannot grow rice in 50 degree normal condition. The maximum is 28.
So can't HSP27 be raised for the other crops also? Say like where you wanted to really work upon all these stress, say salinity stress or drought stress in some other crop species, say like wheat or where you have this kind of problem. Not with even these crops, but the lentils are one of the best possible candidate for transgenics. Because the lentils have many other problems as far as the double stress is concerned. So I think HSP27 would be the best for it.
Yeah. So like I have given this HSP27 to IIT Roorkee, the Harsh Chauhan. So he's work on wheat and barley. So he is now transferring it in the wheat and barley. So by this way, like that's why I'm telling you, this all the underutilized crops are the golden treasure actually. If somebody starts, they will get something in this, because very less research has been done. That's why I always advocate the newcomer people that you should take all the underutilized crop, because in rice, wheat, maize, a lot of people are doing. There is so much competition. You cannot compete. But if you are
sorry to interrupt you, sir.
Yeah.
So we also started work here on the pearl millet. I would like to ask, where are the different institutes which are working on these unutilized crops, so that we can know much better about these crops? Because in India, whatever we know, you are working on this foxtail millet, but we don't know any other personality. which are working on this such type of the scale. So may you help me in Punjab, Rajasthan, Haryana, and even in the Gujarat, Maharashtra, Madhya Pradesh, Chhattisgarh, which institute is working, dedicated working on these crops?
I know in Chhattisgarh that Central University, there Girish Chandel was there. He was
Girish
also
Chandel,
he's a good worker. I can give you the few names. Then IIM, Indian Institute of Millet Research, that in Hyderabad, ICER Institute. They are dedicatedly, they have done a fantastic job for the food processing of millet. They have done an excellent job for food processing purpose. They have developed all the cookies and all the pastas and other thing for from the millets actually, and they are marketing it.
Yes, sir. So it's very noble work that you're doing on neglected crops. So similarly, in the epidemiology side, neglected tropical diseases also is emerging.
Yeah, exactly. Yeah.
NTD. So we have Professor Monisha Dhiman in our university. She's working on leishmaniasis and other neglected tropical diseases, you know. And exactly. Yeah, there are a lot. There is a journal also, isn't it? NTD. Yeah. So now, coming to the question, so we have a lot of questions. So one of the question, I mean, quite similar, a lot of concerns. Manisha Sharma is asking, GM crops either directly or indirectly affecting the biodiversity. What could be the different measures we can develop as to improve the crop quality as well as not harming the biodiversity? Again, the claim is questionable. Is it really harming or not, as you said earlier? Another question is by Devendra Mudgil. Are genetically modified crops good for our environment or not? Over to you.
So whatever GM crops has been released by the government or worldwide, all this kind of study has been done. Otherwise, government regulation doesn't allow to permit to release this thing. But allergy test has been done, and all these biodiversity issues also has been investigated. After doing all this groundwork with 5 or 6 years data, seeing that data, the government or the consultant, the responsible authorities, they actually release the variety. So till date, there is nothing reported that any biodiversity issue with the GMOs. Number one. Number two, whatever trial we have in our country, they are also limited and the confinement zone area. And in outside US, they have several GMOs they are released. In Africa, there are several GMOs released. In our neighboring countries, Bangladesh also has released several. Nepal is also released several. But in India, we eat GMO, but not legally, illegally, because the brinjal and all sorts of maize you are eating, all are GMOs, I can tell you.
We have one question from Durgesh, Khushbu Agarwal. She's asking about the lab to the crops, the field. Hello, sir. How are the things going from the lab to field? I mean, what is the success rate among the farmers for adopting these crops and field trial, and how efficiently has been done?
Okay. For any GMOs or any breeding lines that has been released, there has been gone for all India trial in different location, different environment, and different season for at least consecutive 4 to 5 years. And based on the data, the granting agency approve whether it is good for the farmer or not, because the farmers are the best. Because if you by force, if you release one variety, if it is not performing in the field, farmers will never take. So it is not like this that I can sell something and then I forget this thing, because it is just like the bread and butter for the farmers. So government also has very cautious regarding this, that if the data of yield and stress is not good, they will never release this. Otherwise, it is not good for the government also, because then farmer will never trust the government. So, and based on the data trial, it was released, and it was very actually tough to release any variety at the present context.
One person, Krishan Nanda Ingle, asked a very, very good question regarding the views of the states and the central governments. He asked, how can we efficiently reintroduce these minor and the nutrition millets in nearby villages in the fields actually. So what I would like to add one more things. What is the policy of our governments actually to reintroduce these millets crops in some area, in marginal lands, to get maximum benefits for these crops?
So if you see the marginal area, the local farmers, they used to grow it without any government intervention, because they multiplied their own seeds, they keep it for the next season and they grow it and they consume by themselves. But there is not too much area so that they can sell it to the others. So marginal area, marginal farmers where the lands are not fertile, they used to grow. Still today they are growing. But the question is how, but this is the policy decision should be taken by the government, that they should give something to the farmers so that they can promote the cultivation of these underutilized crop species. Because the corn, millet, and sorghum, these are highly cultivated, not like the other thing. But the small millets, like there are 8 small millet other than pearl millet and sorghum. They're also marginally cultivated in the scattered regions. But government has to take a policy to give the benefit to the farmer, that if they can grow this, the government will buy it, or government will give some subsidies, something like this. Because cost-wise, growing this kind of underutilized crop is not too much, actually, I can tell you. There is no fertilizer required, no water is required. So to grow foxtail millet, you require less than half the water what you require to grow a rice for the same amount.
Okay, we have one participant, Hossein. You may please unmute Mr. Mondal, Hossein Mondal, and you may ask the question.
Actually, I have just one small query. If the neglected crops or the underutilized crop is cultivated without any stress, suppose under ideal conditions, then the quality of the produce will be the same or different?
Look, so all the millets crops are hardy crops. They grow in a harsh environment. Okay, so whether it is a control environment or it's a harsh environment, maybe there is a little difference of production of yield, but I don't think there is much difference, because this is known for its hardy and this stress tolerant.
Actually, I am asking these questions due to some reasons. Because in West Bengal, one basmati aromatic rice is grown in North Dinajpur and South Dinajpur, but they are very neglected crops. But they are these cultivation generally performed in the marginal lands. There is no fertility or irrigation system is developed. But at the end of the seasons, the terminal drought is very important for the aroma as well as the quality. So, but when the farmers is cultivated under good ideal conditions, the same type of flavor and the quality just a little bit different. So, this case study actually drives to ask this type of questions.
Now look,
so is there any study?
During my PhD, I worked on rice and with the West Bengal traditional rice, and that 2-acetyl 1-pyrroline, that the aromatic composition. But these are volatile, huh? So this is aromatic rice is depending on the environment, and the volatile composition is purely dependent on the environment. So
exactly.
So it's not that you get the aroma for each and every places if you grow it. So it requires a certain, like this.
Exactly. Actually, farmer perception is that they are not the similar. That's why there is a issues is coming. That's why I'm asking.
Okay.
Yes, sir. Thank you so much, sir.
Yes,
and because we have already, you spent more than 1.5 hours with us this afternoon. You have committed for only 1 hour, but we have really offshored.
Yeah,
and
yes.
Thank you so much.
It's my pleasure. It's my pleasure.
It's a really great honor for us also to interact with you this long, and you have answered so many questions from us. Sanjeev is also here. Would you like to Sanjeev? I don't see him now. Okay.
Yeah, I'm here.
Yes, Sanjeev. Yeah, would you like to say a few words?
Yeah, of course. It's such a privilege for us, sir, to see you on this platform. Indeed, your presentation, your talk was very brief. I know that so much of work cannot be presented or explained in just 40 or 45 minutes. But the way you presented, I mean, it was so very simple and it was too very understandable. First of all, I would like to thank you for, accepting our department, our university invitation and, sharing your thoughts, your research with us. Sir, I just, it's my personal question. I'm nothing like that. I know you are a geneticist. You're trained and a student of a very renowned person. My question is related to conventional science and this cutting edge technology, that the conventional breeding and what do you say, CRISPR-Cas, the cutting edge research.
Yes.
So where do you think these both technologies they stand? Natural selection versus the synthetic selection, and what would be the future of this CRISPR-Cas? You have explained in depth, but it's just my query.
So still we don't have any policy for this CRISPR genome-edited line, whether it is a GMO or not. But in some European countries and that USA, they have termed as a non-GMO, because here you are editing one gene and not disturbing the other. And then what you have to do is this: once you develop this genome-edited lines, you have to backcross with your variety of your interest. Again, you come into the classical breeding thing. Yeah. So by within short time, you just edit a line to get a desired phenotype, and then with the desired phenotype, you just cross it with your local high yielding or the stress resistant variety, so that you can put it in the breeding line that just integrates that genome-edited line to your breeding line, and by this way you can convert as a non-GMO. Actually. Oh, okay. So that is the way I think the other people, other country, and we are also doing the same way. So once it is the genome-edited line is there, we just crossed it with these our Indian varieties or the varieties which has been cultivated, so that that gene can be introduced and so in the local varieties, so that within 2-3 breeding cycle it is almost it is integrated, and then by this way you can also get the data from this different environmental yield and other stuff to challenge this that GMO that regulatories actually.
Thank you.
Genome editing is very promising, I can tell you. This is like that pre-harvest sprouting I have shown you. It's a very problematic thing, but this one gene QSD1, that addition of 3 nucleotide or amino acid, that changed the whole scenario. Even that domestication of this tomato from a wild to these 6 genes, they have just edited, and then the whole thing is like the wild become domesticated actually. Okay, so this kind of technology, very powerful technology. We have to judiciously use this, and we have to do it for the benefit of the science and benefit of the society, so that we can bring them into our breeding cycle, because there is no alternative of the breeding. I can tell you the classical breeding will be there, should be there, and in future will be there.
It will remain there, yeah. Yes,
it will remain there. There is no replacement of classical breeding. Anyway, whatever you do, but you cannot replace a breeder.
Of course, we will have to end up with the classical breeding.
Yes.
But sir, मतलब मेरा एक concern था आजकल खास करके CRISPR-Cas पे बहुत सारी डिस्कशन चल रही हैं. अब आपका तो बेसिकली एक्सपीरियंस है, मेरा इतना एक्सपीरियंस है कि मैंने पढ़ा है. My basic question comes in basically for the plant species where we are dealing with kind of a gene families where we have a multiple genes and their genes are spread all over the genome. Now, if we wanted to switch off or we wanted to make out or knock out few of those genes or one of those genes, do you think that those particular targets can be managed with the present?
At a time, you can silence 12 or 15 genes. So in brassica, in one of my colleague, Naveen Beast, is working in brassica Nepals. It's a polyploid crop. Okay. So you will get 3 or 4 copies or 5 copies for the single gene. Okay. So he actually did all the silencing for all the copies, exactly what he required, but except this gene where he doesn't want to change anything. Okay, now, because anyway, you have to do sequencing. Once you do this, once you get a line, you will get the answer.
Yes, yes, yes.
CRISPR-Cas9 has the technology to go ahead with the multiple targets simultaneously.
Yeah.
Yeah, no. My question basically was related to gene families, but yeah, I think that is possible, and I think near future this particular technology is evolving day by day, so the things would come evolving. Thank you
Because
so much.
for this technology, designing guide RNA is the basic important thing. Who knows how to design guide RNA, you will get a correct result.
Is it possible that now there can be, say, like 2-3 targets on the guided RNA? No.
Yeah. That's why I'm telling you. Now, if there are several homologs in for this particular gene, you can everyone.
Yes. Yeah. Hanji.
All right, sir. Thanks. Yeah, I mean, it's really wonderful. And on a positive note about this, the coexistence of the modern and classical together, I think we can conclude the session now. And thanks a lot, sir, for sparing 1.5 hours with us. And I hope
thank you very much, sir.
Thank you, sir.
Thank you very much.
Thank you very much.
Thank you.
And now I would like to move on to the next session of the same program. And the next session is about the quiz. It's a biodiversity quiz. So please stay tuned. Okay, let me share my screen. I will now speak a little bit about the rules and regulations and how to participate. Okay, that's really, really important. And how to get on to the quiz. Okay, so the technical side. So in the Department of Botany, this is the 5th major quiz that we are organizing here in this year. 28th Feb, it was a Science Day quiz. 12th March, it was International Quiz on Azadi Ka Amrit Mahotsav. The 22nd April was the Mother Earth quiz, and then 17th April is the COVID-19 quiz. And today's quiz, the biodiversity quiz, I hope you have attended very carefully about Professor Manoj Prasad's talk, because during the talk, I was making the questions. So, you will see that some of the questions are from his talk. So, it will show you, are you really attentive or not? So this quiz is, the instructions include, it's open for everybody, including the teachers, school students, foreign nationals, unemployed youth, everybody, it's completely open. And winners, the first 3 positions will receive certificates. And there is no cash prize. Mind it, there is no cash prize, but we are issuing the certificates. Of course, it's highly reputed, it's valuable certificate by the university. And the quiz is conducted by menti.com website. Mind, if you're watching in the YouTube, stay tuned till I complete that instruction. And after that, you need to go to this particular site, menti.com. Many of you have attended many of our quiz, you know how it works. But if you're new to this particular quiz, mind that in the YouTube, like in the chat box, answer A, B, C will not fetch you any point. You would need to go to this particular site, menti.com. You can just write on a piece of paper. This is the site name, menti.com. Okay, M for Mumbai, E for elephant, N for new, T for Tokyo, I for India.com, menti.com, right? And yeah, or Zoom also will not fetch you anything. So when you go to this site, of course, it will ask you the code. I'm going to tell you the code. And it will also ask you to enter your nickname. And remember, don't add your nickname. It has to be your real name. Otherwise, we will have a hard time to understand who is that nickname belongs to what. Like if you choose a cat or a dog, so to whom that belongs to, we don't know, right? So make sure you're entering your real name, okay? So nicknames are not accepted, right? So the quiz has got timed questions. It's something like Kaun Banega Crorepati, the Bachchan's program, and so many of its reincarnation in the TV programs. So it has got timed questions, and when the quiz starts, the question will appear, and the participants have to answer the question before the countdown. But as fast as you answer, you will get more points. That is the trick of it. And if you answer it wrong or no answer, you will get no points. It's a 0 point. So you can guess, no problem. The faster you answer, the higher score that you're going to get. So the program even measures the response time even up to microseconds. So looking up for the answer in the internet is not a good idea. Googling for the right answer and choosing the right answer, you're simply losing all the points. Make sure you're actually responding faster. And after every question, there will be a leaderboard that will automatically declare who answered faster and who is getting the top score till then. So everything is automatic, so we don't have to work on it. And at the end of the last question, the winners are declared. And after the quiz, please join back here in the YouTube. Okay, so because you're watching the YouTube, I repeat, in the YouTube chat box, the answers will not be considered for declaring who's the winner. So you would need to go to the menti.com. And if you are in the Zoom, I can see very less people only in the Zoom, no problem. But do not speak the right answer, okay? And do not annotate in the Zoom. Some mistakes that over the last many quiz competition, I've seen that some participants do annotate in the Zoom. Please refrain from it. Now the quiz is going to start in 4 minutes. Please go to the site menti.com and enter the code 102624. So you would need to open in any standard browser. For example, if you're watching this in the laptop, you can open it on your mobile phone, any browser, Chrome or Firefox or whatever, Edge or Microsoft Explorer. Go to the site menti.com, and when you enter, you will be asking, the site will be asking to enter the code. The code is 1026204. I repeat, 1026204. So please go to the site menti.com, right? MENTI.com, and enter the code 1026204. And once you enter this code, it will ask you your nickname, and do not give any nickname. So instead, you present your real name. Okay, so that is what. Let me go to that site once again so that I can see how many are here online. So by the way, Professor Manoj Prasad's talk, there were more than 650 live views. Of course, a lot of people are going to join and watch the program because it does automatically archived in the university's official YouTube channel. So many more are going to watch it. And now I can see lots of participants already in this menti.com. We are beginning shortly in 3 minutes. Okay, please stay tuned. I repeat, if you are watching this in the YouTube, you have to open a separate tab and you have to go to a site called menti.com, MENTI.com, and use the code 1026204. All right, 1026204. And we are going to begin the competition very soon. It's only a buffer time because so many people are still going to that particular site. And make sure that you have very good internet connectivity. And especially if you're in a village, I suggest you to go to the terrace or out to your garden so that the receptivity is pretty high, So we are not responsible, right? If the internet is broken. So anyway, it's like a infotainment, right? This is not like winner, who is a winner and loser. It's the quiz is part of our outreach activity and it's part of education, So it's an infotainment. You're learning many new things from the quiz. So take in that spirit. It's part of the Central University of Punjab's outreach activity. We can see a large number of participants now, and the last time I'm repeating. Please go to menti.com, MENTI.com. And when you go to that website, it will ask you the code, and the code is 1026204. 1026204. And it will ask your nickname, add your real name. No nicknames are allowed, okay? I can see participants are increasing. Let me wait for a few more seconds before we begin, okay? So yeah, I can see that lots of participants are joining in our program. And I'm really happy because the quiz competition, I added it very late. Earlier, I did not announce, and still I can see a large number of participants coming for the quiz. Even though there is no cash prize, I would like to highlight it because participants are not after only that kind of goal, right? Materialistic goal. It's a privilege. And friends, it's not about winning or losing. It's more about learning, right? Let us learn everything. Something let us learn every single day. So that is the spirit of this quiz competition. I can see a very large number of people already. And now let us begin the quiz with our first question. So stay tuned And yeah, within few minutes, I'm going to start, with few seconds, I'm going to start with the first question. All right, let's begin, right? And make sure these quiz, there is a timed, okay? The response time, we are measuring in microseconds. So faster you answer, more points you're going to get. So let us begin with our first question of the day. Answer fast to get more points. Tardigrade, sometimes referred as the toughest organism on the planet Earth, is insect, plant, bacteria, virus. Yeah, most of you answered it insect, and yes, it's the right answer, insect. You know, the tardigrade is also known as a water bear, isn't it? So it's one of the cutest animal. You can see it everywhere, even in Antarctica, Arctic, you know, in desert, everywhere on the planet Earth. And maybe if I want to put one organism for the UN, you know, World Biodiversity Day, it could be tardigrade. Even in the space, you can find this organism. It's really tough organism. You see that it's an insect. And next question. And now at the end of the first question, who is topping here? I can see some familiar names. Some of our students, I can see Deepak Das is the fastest, followed by Malay Kumar. Okay, so let's see if they can able to see, get these leads on. Go into the second question. Question number 2. Which among the following is the most threatened species? In the biodiversity conservation point of view, the most threatened. Asian elephants, monitor lizards, olive ridley sea turtle. Most of you answered olive ridley sea turtle. No, you know, it might surprise you. You know, that is the kind of questions I really love in this the quiz competition. Sometimes what we believe is completely wrong. Asian elephants. Friends, elephants is endangered and it's enlisted in the IUCN Red Data book. While olive ridley is only vulnerable, you know, it's 2 steps behind the endangered. Endangered is really, really threatened, you know. And while monitor lizard is least concern, LC. These are the, you know, the flags of the Red Data book. So Asian elephant is the right answer. It's highly endangered. So at the end of the second round, let us see the leaderboard. So we can see that Bodhu Tejaswini is coming on the top after the second round. I can see that Tejaswini, that name is very familiar because she's one of our old student, because we pinged in our alumni group also. Yeah, and now let us go to the third question of the day. Scientific evidences such as that novel coronavirus relative has been circulating for decades unnoticed in which of the following animals? Giant panda, horseshoe bats, pangolins, Chinese dugongs. Horseshoe bats is the right answer, and yes, most of you got it right. Correct. It's a horseshoe bats. It's not exactly the novel coronavirus, but the closest relative, you know, the Coronaviridae family members. That is what the new evidences point to. Now, at the end of this round, let us go back and check the leaderboard once again. Is Tejaswini still on top? I think Tejaswini couldn't get it. Deepak Das is now the fastest one. And yes, and followed by Smritiksha Mohanty. Now, the next question, fourth question here. Which of the following has the largest genome in the world? Lungfish, chimpanzee, human beings, Pandora virus salinus. So this talk is about the genomics, isn't it? So this is about the genome size. Friends, it's not the human beings. Most of you answered human beings. The human being, the genome size is 3.3 gigabases. At the same time, it is the the lungfish is the biggest. The lungfish, its gigabase is 130 gigabase. The largest one is the lungfish. And we are learning these things every day. The scientists are learning new information, isn't it? So that is what is happening now. At the end of this round, the 4 questions, we can see that. Who is topping this list? Smritiksha Mohanty. Yes, so Smritiksha is topping this. Let us see that. It's only the 4 questions, right? Many questions are to be gone. Now, the 5th question, we have 15 questions, okay, in this quiz competition. The 5th question is, which of the following animals can detect smell hundreds of kilometers away? Really good olfactory sense. Cats, dogs, porcupines, bears. Yeah, it looks like neck-to-neck fight, dogs and bears. Bears is the right answer, grizzly bears, friends. I also learned it only very recently, so I thought the dogs are really great sniffers, but no. Grizzly bears can detect the smell even 32 kilometers away. You see, 32 kilometers, a big thing. And their olfactory sensation is 2,100 times than that of the human beings. So it's really, really powerful. The grizzly bear, the brown bear. So at the end of 5 questions, going back to the leaderboard, Anupriya Yadav is now coming on the top. Very interesting. Good job, all you guys. So stay tuned for the next 10 more questions. So now 6th question. Arctic tern in its lifetime flies a distance equivalent to 3 times a trip to the moon and back. True or false? Yes, it's true. And most of you got it right. Friends, if you ask me which is my favorite animal, I would say that Arctic tern. It's such a small animal, friends. It's only 100 grams. And do you know how much it can actually go from Arctic to Antarctic and back every single year, friends? And that means 80,000 kilometers per year. And the Arctic tern can live up to 30 years. So it's totally 2.4 million kilometers, just 100 gram. Look at that, the power of nature. Can you ever imagine this kind of a flight or a drone, just 100 gram that can achieve it? It's amazing. I've seen Arctic tern during my voyage to Antarctica. Very interesting. And now going back to the leaderboard once again. Parul Kumar got it right first. And who is topping the list? Very interesting. Now Parul Kumar is charting the chart, followed by Smritiksha Mohanty. Now the next question. Question 7. Humans evolved from monkeys. True or false? So most of you answered it true, but no, that is incorrect. That's a misconception. It's a very common misconception about those who are not really understanding how the evolution works. None of the two extant, that means living organisms, have been evolved from one another. We can only say that humans and current day monkeys have a common ancestor. Humans and chimpanzee have a common ancestor. We can never say that we evolved from chimpanzee. No. So that is not the right way to interpret it, So that is why it is a false. It's a very common misconception. So at the end of 7 rounds, let's go back to the leaderboard and see once again, Abhijit and Rupak did a fast response at the end of this round. We can see Rupak Kumar is now charting, followed by Deepak Das and Malay Kumar. Very interesting. Now, 8th question. Oldest crop cultivated in human history, millet or rice? Yes, millet. I am very happy. Most of you got it right. Yes, this is the question directly taken from the presentation of Professor, our chief guest here, Dr. Manoj Prasad. And it is, he has just explained that it's over 4,000 years older than, how old the rice is. Right? The millet is really old, one of the oldest crop in our human history. And at the end of this round, we can go once again back to the leaderboard. We can see that Deepak Das is still topping, followed by Malay Kumar and Abhijit Nayak. Now let us go to the next question. Question number 9. Adenovirus in the Covishield vaccine originally comes from which? In the Covishield, the coronavirus vaccine, which animal? Guinea pigs, chimpanzee, cow, or bats? A less known fact. So most of you said bats. Incorrect. The right answer is chimpanzee. Friends, this particular vaccine, Covishield, its actual name is Chadox and COV2. So the Chadox stands with CHA. That is basically, the AD stands for adenovirus. CH stands for chimpanzee. CHAD adenovirus. OX stands for Oxford University, where it is originally developed. Covishield. Now in India, of course, it is by SII in Pune, and we have another vaccine, Covaxin. So these two are the vaccines. So it's basically from chimpanzee. The adenovirus in the chimpanzee has been recombinant DNA, the genetically modified, just like how the Dr. Prasad explained today. So that is how the vaccine formed. Going back to the leaderboard after the 9 questions, we can see that who is topping the chart. Malay Kumar looks like he's, and Jay Krishnan VM, I can see a new entry here. Deepak Das, Malay Kumar, and Abhijit Nayak still topping. Now, 10th question of the day. Which among the following is not a biodiversity hotspot? Professor R.K. Tiwari sir. R.P. Tiwari sir has said about the biodiversity hotspot early on. Western Ghats, Himalayas, Aravalli Range. Which is not? That is the question here. Aravalli Range is the right answer. Friends, India has got 4 hotspots, just like our Professor R.P. Tiwari sir has just said in his the inaugural address. These are basically the first 2, that is Western Ghats, Himalayas, plus Sundaland, that is where the Andaman Nicobar Island is in, and Indo-Burma region. And these 4 are our hotspots, the biodiversity hotspots. Going back to the leaderboard, we can see that Bharati and Karavadra are some of the fastest respondents, and still the topper is Deepak Das and Malay Kumar. Now, 11th question. Which among the following is a marine national park? India has marine national park, Wandoor in Andaman Nicobar Islands, Gulf of Kutch in Gujarat, Gulf of Mannar in Tamil Nadu, all of above, none of above. All of above is the right answer. Most of you got it right. Yes, we have 6 marine national parks in India, friends. Marine national park, not I'm saying about the normal national park. And yes, all these are rich in marine biodiversity. And now the next question. Before that, let's go back to the leaderboard once again, and we can see that Jay Krishnan VM is now coming on top. And still, we can see that Deepak Das and Abhijit Nayak are first and second positions. Question number 12. Only 3 more questions left. Lungs of planet responsible for more than 65% of oxygen in the atmosphere. Oceans, trees, especially tropical rainforest. Most of you answered tropical rainforest and trees. No, the right answer is oceans. And that is also very interesting things that most of us don't know about the significance of oceans for the primary production. Friends, 65%, more than that, it's basically 2/3 of the oxygen in the air that we breathe comes from the oceans by phytoplanktons, especially 2 genus, Synechococcus and Prochlorococcus. So it's one of the less known fact, highly inconspicuous. Even the syllabus, the botany syllabus are not teaching, so this particular thing all over the world. There is a lot of bias for the terrestrial, terrestrial plants, right? The marine plants are really, really important. Oceans do produce a lot of oxygen in the air that we breathe. So going back to the leaderboard, Rupak Kumar answered it very fast. And looks like the topper is still same. Deepak Das with 9,250 points is topper, followed by Abhijit Nayak, and Bharati is now number 3. Bharati's score is 8,679. 2 more questions to go. 13th question. UN International Mother Earth Day is celebrated on which day? 5th of June, 22nd April, 22nd March, 3rd March. April is the right answer. I've said that, we organized a quiz on that day as well. 22nd April is UN International Mother Earth Day. And look at the word choice the UN has used. It's not Earth Day, it's called Mother Earth Day. Friends, respect the Mother Earth, Prithvi, our own mother, you see. We have to treat her respectfully. That is the message, the key message of this UN's International Day celebrated on 22nd of April every year. So looks like same, the leaderboard. Deepak Das couldn't answer it. So now look at that. We can see that the fastest is Abhijit Nayak is now on top, followed by Bharati and then Deepak Das. Look at that. One question can completely change the leaderboard. And now coming to 14th question. Only 2 more questions to go. UN World Wildlife Day is celebrated on which day? 5th June, 22nd April, 22nd March, 3rd March. Third March is the right answer, and most of you got it right. Yes, 3rd March is celebrated as UN World Wildlife Day. All these day and observations are connected, interrelated, because all these are something to do with environment, sustainability, and biodiversity. All these 4 or 5 days of the UN International Observances. Now, going back to the leaderboard after this penultimate question, we can see that Abhijit Nayak got it faster. Naman Gupta and Abhishek Kumar Singh, they are the fastest respondents. And Abhijit Nayak is still topping, followed by Bharati and then Deepak Das. Now coming to the final question of today's, the quiz competition, 15th question. Caffeine is coffee plant's natural defense mechanism. True or false? So most of you said true. Yes, it is the right answer. Friends, what coffee that we drink, the caffeine in the coffee is basically a defense mechanism, a herbivore, a deterrent mechanism of the coffee plants. It leaches into the surrounding soil. And, as it leaves from the plant drops, as the seeds drop in and decompose into the soil. And since caffeine is toxic to other plants, it prevents other plants from growing around the coffee plant and competing for the sunlight. You see, it's a very good adaptation of these coffee plants. And by the way, because of the climate change, it's threatening the coffee cultivation around the world. It's predicted that by 2050, all the good coffee plantations will go. They cannot cultivate the coffee plants because of the global warming. So we really have to take care not to actually harm our Mother Earth. So now that the questions are over, and we'll go back to the final leaderboard. Who is going to be the winner of today's quiz competition? We can see that the winner here is Abhijit Nayak. Congratulations, Abhijit Nayak. 11,723 points. Great job, Abhijit Nayak. Followed by Bharati. Bharati gets 10,396 points. If the name is right, then Bharati, I remember her. She was one of our old student and alumni. Yes. And followed by Deepak Das. Deepak Das get 10,209 points. So these three are the winners. So allow me to snip this particular page for me to issue the certificates to the winners along with the points. Great job, all of you, Abhijit Nayak, Bharati, and Deepak Das. And thanks so much for watching this quiz competition. And I hope you learned something new. That is how it's not about winning or losing the competition. It's about picking up some new knowledge, isn't it? So thanks for watching, and I really enjoyed having this quiz competition, though it is only 15 questions. So yeah. And now I can see a lot of people watching this event. Please come back to the YouTube. So we are approaching the final concluding session of today's quiz competition. And sir is already here, Dr. Manoj sir, Professor Prasad. You may please congratulate the winners. I'm really happy you are with us throughout this competition. Yes, over to you, sir.
I would like to congratulate the Nayak, Mr. Nayak. Thank you very much. Congratulations.
Yeah, thank you, sir. Thank you so much. Now, I would like to invite Vinay, Dr. Vinay, for word of thanks, please.
Yeah, thank you, Dr. Felix. Yeah, under the supervision of Professor Raghavendra Prasad Tiwari, Honorable Vice Chancellor of our university, and with the guidance of the Dean Academic Affairs Ramakrishna Vujrika, HOD Felix, and on the behalf of all these persons, my faculty, senior persons, Professor Sanjeev, Dr. Pankaj, and me, and all the listeners who attended this meeting talks of the Dr. Manoj Prasad. So I would like to express thanks each and everyone. And I don't want to forget to express the thanks to lab number 103, NIPGR. All the members of the 103, as well as all those who are here. I know Dr. Felix Manoj Prasad from the last 7 to 8 years. He is a very charming and energetic person. You were saying, and still in this age, he seems to be a young, just like a 25 years old. And he's very fond of foods also, as I know about him. And all those persons who are fond of foods are very pure from hearts actually. So thank you, Manoj sir, you and all the members who attended this meeting. And I would like to express my sincere thanks to Tiwari sir also to grace this meeting and attend this meeting from the very first minutes and up to here. So thank you each and everyone. Yes, Dr. Felix.
Now, the final program. This is a national program. This is, this is what we do with every program. Please raise seats for the national anthem. Jai Hind. Thank you so much.